Hello everyone, and welcome to today's webinar, Time-Resolved Electron Microscopy, Synaptic Ultrastructure, Millisecond by Millisecond with High Pressure Freezing. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. This educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd now like to welcome our speakers, Dr. Shigeki Watanabe, Associate Professor of Cell Biology at Johns Hopkins University, and Grant Kusick, Ph.D. candidate at Johns Hopkins University. Shigeki, Grant, you may now begin your presentation.
Okay, so thank you so much for such a nice introduction, and thank you all for tuning in today. I'm Shigeki Watanabe, and I'm an Associate Professor in the Department of Cell Biology at Johns Hopkins. And today, with my graduate student, Grant Kusick, we are going to talk about high pressure freezing and how we are using this technique to basically look at membrane dynamics at the synapses using electron microscopy. And so this talk is divided into two parts. The first part is going to be taught by, it's going to be me talking about why we are using electron microscopy and then why we have to use high pressure freezing technique. And the second part, Grant is going to talk about how we are adding the temporal dimensions into electron microscopy, and then how we are using this technique to basically looking at how synaptic vesicles actually move at the synapses. And so, so let's get started. And so I'm going to start talking about why electron microscopy. And so, as you know, seeing is believing, and then this is totally true. And so if you just look at this electron micrograph, this is just so beautiful, right? You can see how the nucleus is organized in the cell, and then how they're embedded within this endoplasmic reticulum. And you can see the organization of all these things that are found within the nucleus, such as nucleolus, or you can see heterochromatin organization, or even the nuclear pore that you can see in this picture. And so this is just such a beautiful image. And then now, with over the last 10 years, there was a big development in a correlated microscopy technique that allows us to actually pinpoint where the proteins are in these images as well. And so you can really localize where the proteins are to the subcellular structures that you can actually see in electron micrograph. And so this is one of the example images that we had back in 2011, basically coupling super resolution microscopy technique with the EM to basically look at where the histone molecules are in this particular case. So you can really see where the heterochromatin organization, where the heterochromatins are at the nucleus. Electron microscopy is also great for looking at the protein complexes, and so you can look at these sensory neurons in C. elegans in this case, and you can see these microtubules, and then you can see how the protofilaments are actually organized in this particular image. And then, as you know, that the protofilaments, these are basically looking like this. And then if you had to do a 3D reconstruction of these images, what you're going to see is that there's a nice, you can even look at the protein complexes from these images as well. And so electron microscopy can really get down to the protein resolutions, especially with the cryo-electron tomography these days. And then electron microscopy is also great for what we are interested, for looking at what we are interested in, and this is the synapses. And so this is an example image from ventral nerve cord of C. elegans, and you can see this is the ventral nerve cord over here, and then there's also a muscle on the right side of the picture. And then what you see here is that there's actually a motor neurons that controls this muscle movement. And then if you zoom into one of these images, this is what we are interested in looking at. And so here is the synapse, and these are neuromuscular junctions, and you see these circular structures within this membrane, within the membrane. These are called synaptic vesicles. These contain neurotransmitter, and then they are used during the neurotransmission. And so what you can see here, as noted as docked synaptic vesicles here, they are actually touching the plasma membrane. They are in a cross association with the plasma membrane. And then this actually cross association is quite important for its function. And there's actually a vesicle next to it, which actually has some distance to these plasma membrane. These are also important, but this 5 nanometer difference between these vesicles and the vesicle next to it, to the plasma membrane, is actually quite important for how synaptic transmission actually happens. And Grant is going to talk more about this topic. But as you can see in these images, electron microscopy can allow you to look at these organelles that you cannot see otherwise. And so electron microscopy is really good at looking at structures within the cell. And so to summarize, what electron microscopy is good for is really looking at the membranes and the organelle organization, and then looking at the protein localization below the diffraction limit. I told you about the Fourier microscopy, but there's also immunoelectron microscopy that you can use. And then this is also good at looking at the structure of protein complexes, especially with the cryo-electron microscopy these days. But what's not good with the electron microscopy is to looking at the whole cells or tissues. I mean, it's possible, but this actually requires reconstruction, which is time consuming. And then this is also not good for looking at the live tissues or looking at the change in the membrane morphology over time. And Grant is going to address how we can actually look at the membrane dynamics on the millisecond time scale by using a technique of flashing freeze or zapping freeze. Okay, and so I hope that I was able to convince you that the electron microscopy is really great at looking at the structures and things that we care about. But why do we have to use the high pressure freezing? And so the reason for this is quite simple. This is because we are using electrons as a source of imaging. And so here's the tissue. The electron microscopy kind of works like the X-ray, or we have broken bonds, for example. And so we are basically trying to cast the shadows of electron-dense materials so that we can image this whatever biological tissue you have on the film or the camera. Right? The problem with the electron, though, is that electron can get scattered with any molecules that you have on the path. And so any, for example, water molecules that are in a column, this is going to interfere with the incident electrons, and then therefore you cannot have electrons that can be used to image your tissues. And so in order to avoid this, electron microscope is operated under the vacuum. This is actually bad for biological tissues because biological tissues are usually hydrated. And what this means is that you have to actually dehydrate tissues in order to image them under the microscope. And so you can think of a situation like a grape and raisin. And so if you dehydrate a grape, basically it's going to look like a raisin. So as you can see, the morphology of grape is totally distorted in this case. And so in order to avoid this, what people typically do is to fix structures using aldehyde-based fixatives, right? And so you can actually cross-link tissues, cross-link molecules, and so that you can retain the certain structures. But this is still not good enough. And so in fact, this example from Graham Knott's lab, basically looking at the brain slices from mouse, what you can see is that the fresh tissue, comparing fresh tissue to the chemical fixed tissues, you can see the size difference. You are basically shrinking tissues basically by just chemical fixing tissue. And so this is still not good enough, right? And so what you want to do is to be able to retain tissues in its original morphology. And so the only way to get around this is basically freeze those tissues so that we can actually keep them in the original form. Freezing tissue, though, is going to be a little bit of a problem. The reason for this is that, as you know, the water molecules, when you try to freeze, what's going to happen is that it's going to form a hexagonal ice crystals, and then they basically expand, right? And so when the ice crystal expands, then what's going to happen is that it's going to basically break the tissues apart. And then also another thing that happens here when the ice crystal forms is that all the solids, like proteins and salts, is going to be excluded from the ice crystals, such that the local osmotic stress is going to be different. And this also basically can break the tissues apart. And so because of all of this, you have to avoid ice crystal formations. And then in order to do this, there are a few ways that that we have come up with, like what I mean by we, the researchers over the 5 decades, basically, came up with is basically the way to freeze tissues without ice crystal formation. And then there are largely 3 ways to freeze tissues without that ice crystal formation. The first technique is kind of like making an ice cream. Basically, you have to add cryoprotectant into the tissue. And so you can nuke tissues with sugar or milk for like making ice cream. Basically, like you can add a lot of sugar and then preserve tissues. And so when you eat ice cream, there's no this crystal-like texture. It's because there's no ice crystals forming in ice cream. And so you can do that in the biological tissues by basically nuking tissues with sugar, and this is called Tokuyatsu method. And people use this for doing the immunoelectron microscopy. The second way to basically avoid the ice crystal formation is to use the basically it's kind of like making a dipping dots, where you reduce the volume of the tissue that you're freezing and increasing the freezing rate. And so, as you know, the dipping dots is made by liquid nitrogen by basically dipping small bits of ice cream. And then, although this doesn't have a milk in it, you still don't have this ice crystal, the crystalline-like texture when you eat the dipping dots. And people use this technique for basically single particle cryo-EM or cryo-electron tomography these days. This is called crunch freezing. And so we are basically putting the tissues or samples into liquid ethane in those cases. The third way to get around this ice crystal issue is to make the water sluggish. And this is the super cooling effect that we are talking about. And this is what the high pressure freezing actually does. And so the way that the high pressure freezing actually works is basically making the water sluggish so that they don't have time to actually rearrange into the hexagonal ice as you freeze tissue, so that the water molecules basically stays where they are as you freeze them using liquid nitrogen. And this is what we are actually doing by applying high pressure onto the tissues, 2,000 atmosphere pressure, using in this case, like a high pressure freezer. And then what we do is we come in with the liquid nitrogen to basically freeze tissues instantaneously. And then with this approach, we can retain the tissue integrity without basically compromising the morphology. And so here is an example image. This is the same C. elegans ventral code that I showed you earlier on the right side with the high pressure freezing. But if you look at the conventional aldehyde fixed tissues, what you see is that there's actually all these tissues are there. But if you look at the morphology, the neurons are more like jigsaw puzzle. And if you look at the mitochondria in muscle tissues, their membrane is kind of like ruffled. And so the morphology is really different from what you see on the right. And this is also true in the mouse brain tissues. This is an example image from Graham Knott's lab again. And then what you can see here is that what you're losing in these tissues is this extracellular space. And so normally in the brain tissues, there should be a lot of extracellular space. But when you're using a chemical fix, you can see that there's not much of the extracellular space. Using this technique, using a high pressure freezing technique, we can also preserve protein localization or the structure of membrane much better. And this is actually recently demonstrated by Harold Hess's group using cryo-fixed versus chemically fixed tissues of endoplasmic reticulum that they are looking at here. And as you can see, if you do even a fluorescence microscopy, you can tell that the chemical fixation is totally changing the morphology of all these organelles that you want to look at. And so high pressure freezing basically prevents or avoids all these artifacts that I described and you can retain the morphology of the tissues. And so this is why we want to use high pressure freezing, and then this is actually quite important for looking at the membrane morphology using electron microscopy. And so now I'm going to hand this over to Grant Kusick, who is going to talk about how we can actually add the temporal dimension to electron microscopy, because all of these samples that you are looking at is basically a static image of tissues that are fixed or frozen. And so, Grant, take it over.
Thanks very much, Shigeki. So yeah, I'm Grant, a Ph.D. candidate in Shigeki's lab, and I'm going to talk about how we can combine the nanometer resolution of electron microscopy with millisecond precision of high pressure freezing to look at membrane dynamics at synapses. So in the Watanabe lab, we're very interested in synaptic cell biology. And so Shigeki already showed you an example of this beautiful structure that we see. And just looking at a synapse, you can sort of understand why this is a structure that is traditionally in the domain of electron microscopy. And that's really just because everything is very, very small. Here, looking at it, the entire presynaptic terminal is only about a micron across. Synaptic vesicles are 40 nanometers in diameter and tightly packed together. And all these sort of fine structures of synapse vesicles getting close to the active zone, vesicles fusing, things endocytosing in the presynapse. All this is at a nanometer scale, really only accessible to electron microscopy under most conditions. But the issue here is that, as Shigeki said, electron microscopy is really good and only really good at looking at dead tissue and not at dynamic processes. But the synapse is not only obviously quite dynamic, but operating at a time scale really not seen anywhere else in biology. So we're operating at a spatial scale of nanometers, which is good for EM, but we're also operating at a temporal scale of as fast as milliseconds. And so what we've wanted to do, and what we do in the lab, is combine temporal resolution with spatial resolution to capture these events using high pressure freezing. And so in principle, with high pressure freezing, this is very possible because it intrinsically has to be a fast process. That's part of how you get your beautiful structure preserved. So the EMICE, the current Leica instrument that we use, your actual control of freezing is down to the millisecond. So the high pressure freezing there, in addition to giving you native structure, gives you some of that temporal precision by itself. So what you need then, the next step you need to look at these synaptic processes is a way to precisely stimulate the cells, to couple that stimulation, that induction of action potentials and activity with your freezing. And so the way that Shigeki and others have developed this starting over 10 years ago is initially using a system that they named flash and freeze. So the freeze obviously comes from high pressure freezing. The flash comes from the fact that you're using light to stimulate the cells. And so in this system, they use a molecule that will be familiar to any of the neuroscientists here, channelrhodopsin. So at the time this was developed, channelrhodopsin was a hot new technology in neuroscience, which is widely used because it allows you to, by expressing it in neurons, you can make excitable cells like neurons excitable with light. Because channelrhodopsin is a light-gated cation channel, and that means if you express it in a neuron and then you shine light on the neuron, that channelrhodopsin will open, sodium ions will flow in, and that will trigger an action potential. So if you can think about how you could set this up for your high-pressure freezing experiment, you have your cells already moved into the high-pressure freezing chamber. All you need is a little LED light shined in. So in this case, you shine a LED light for 10 milliseconds, and that usually triggers a single action potential. When that happens, you get what you want. You get activity, you get an action potential that will flow into the synapse, and then trigger all the kind of things you want to see. Vesicles will fuse, release neurotransmitter. It will be received by the postsynapse. You'll see endocytosis, all the kind of things we want to look at. And then at exactly the time point you want, where you think you're going to see the thing you want to see, you freeze the cell, immobilize it immediately. And then once it's frozen, everything is frozen in time essentially forever. And then you can go look at what it looked like in that instant by electron microscopy. So this is just showing an example of essentially how you set that up. All that fancy work of things being very fast and, the stimulation, all that, that's all done by the machine. All we really do is set up your sandwich of solution that goes around the cells, close the cover, and then the machine takes over everything from there. Again, flashing the cells and then freezing them. So it's all just based on what you've programmed into the machine. So all the data I'm going to show you today are all from the main model system we work on in the Watanabe lab, mice, and specifically dissociated mouse hippocampal neurons. But I do want to note that these sort of procedures are applicable and have been applied by Shigeki and others to a variety of different model systems in neuroscience. So essentially anything that's thin enough to high pressure freeze, which is quite a lot of things. So organotypic or acute slices can be done with this, as well as small entire organisms like C. elegans or fly larvae. So when we do flash and freeze, we can see some cool stuff. So we can actually capture what's going on at the synapse during neurotransmission. So for example, you can actually see synaptic vesicles, this process that happens in hundreds of microseconds to milliseconds, these synaptic vesicles that have fused with the plasma membrane and are now having their membrane collapse into the plasma membrane. Critically, what Shigeki discovered in his big breakthrough that was enabled by this technology is he found an entirely new pathway for endocytosis that, because it happens on the scale of hundreds of milliseconds, it's by far the fastest form of endocytosis that occurs in any cell, and it's, as far as we know, unique to synapses, was termed ultra-fast endocytosis. So this advance in technology with flash and freeze and high pressure freezing really has blown the entire field of synaptic vesicle endocytosis and sort of completely redefined the dogma and rewritten the textbooks. But what I'm going to talk about for the rest of today is my focus, which is on synaptic vesicle docking and exocytosis, and how some of that work has been enabled by the next step in the new generation of time-resolved electron microscopy, zap and freeze. So in Shigeki's experiments, I showed how you can see synaptic vesicles that have fused. So it's always been considered in the field that the vesicles that are available to, so a presynaptic terminal is filled with all these hundreds of vesicles, but the ones that are actually ready to fuse are what are shown here as docked vesicles. So docking is, again, something you can only get by electron microscopy. This is a strict morphological definition of a vesicle that is in contact with the plasma membrane. So here, Shigeki in this micrograph is showing the very strict definition. If you're only a few nanometers away, that vesicle is not docked. It's really only those vesicles that are in contact. Again, something you can only see by electron microscopy with cryofixed tissue that are docked and actually ready to fuse. So Shigeki was the one who actually showed that this prediction is really correct, because when you stimulate, you can see that when you get exocytosis, only these docked vesicles are depleted. So these slightly further away tethered vesicles are not. It's really the docked vesicles that fuse and get used up. So this is actually quite a potent effect just from this one action potential. So after only a single action potential, almost 40 to 50% of all your dot vesicles across all your synapses are gone. This is consistent with sort of the thinking about synapses, which is that it's well established that as you stimulate a synapse, they get exhausted. They do what's known as depression. And this all makes sense together because as you're stimulating, you're using up dot vesicles. There's less substrates for neurotransmitter release. You're going to release this neurotransmitter. The synapse is going to depress. And Shigeki showed that synaptic vesicle docking recovery occurs on about the time scale of seconds. So in these preparations for vesicles to get fully back to baseline takes about 10 seconds. So this generally gels with what people know about synapses. You know, vesicles get used up, they get weaker as you stimulate. But any physiologist will tell you that actually many synapses on very fast timescales, on these millisecond timescales, actually increase their outputs and get stronger. And so here, looking at typical electrophysiology data, looking at the outputs of the postsynaptic cell, this is a very characteristic behavior of many synapses, where if you pair two stimuli very closely together, so you stimulate once and you get a certain response, and say you stimulate again 10 milliseconds afterwards, much of the time you actually get a much stronger response from the synapse. So it's actually at these closer time points to your first stimuli, you get your strongest responses, which is weird because those are the time points where you've lost these vesicles and they haven't recovered yet. They take way longer than this to recover from the data that we have. So the way people normally think about this in the field is that, all right, you have fewer vesicles there, but somehow the vesicles that are remaining become more likely to fuse. So you're essentially making up for this lack of vesicles with sort of the remaining ones picking up the slack. There are a lot of limitations to this idea, not least that as you have fewer vesicles and the vesicles remaining are more and more likely to fuse, you would expect the synapse to just really exhaust itself out, which again, they don't do. Many synapses can keep firing for a long period of time. So what we thought about is, I mean, it's in principle a very simple idea. Well, what if you just have recovery of vesicles that's quicker? You have vesicles that dock in really quickly. But again, this isn't what Shigeki saw with his experiments, right? Docking took about 10 seconds to come back. But here again, we come to a technical limitation, which is that while flash and freeze by itself is really impressive, at this time scale, actually, there could still be things we're missing. And that's because while the high pressure freezing, as I said, is extremely precise down to about the millisecond, channel rhodopsin itself as a tool has a lot of limitations and potential artifacts you introduce. The most relevant one to this being that your temporal resolution is somewhat limited. I said we had to shine a light for about 10 milliseconds. The action potential can fire any time within that 10 milliseconds and even afterwards. So essentially, we're limiting our temporal resolution down to 10 milliseconds. So within all those precise, delicate events that are happening over the course of milliseconds, we really could be missing those with this technique. So to sort of overcome this obstacle, Wayne Davis and the engineers that like it came up with, in principle, a very simple solution, which is, well, why don't you just electrically stimulate the neurons? That's what they normally respond to. They're electrically excitable cells. So the problem here is that shoving a bunch of wires into a high pressure freezer that's then going to go to 2,000 atmospheres is not a good idea. So the solution they came up with is to use the same light stimulation system, but instead of having the light stimulate the cells, you have the light trigger a photoelectric circuit. So here's sort of the basic setup of what we term zap and freeze, and the principle by which it works. Essentially, you just have a capacitor that's charged up and closed by a photoelectric switch. So when this is charged up, the neurons aren't being stimulated. You charge it up, everything goes into the high pressure freezer, and then once in there, everything works just like it does in flash and freeze, except now your light shines onto that photoelectric cell, that closes the circuit, and then you just shine the light for a millisecond, boom, those cells get zapped for a millisecond. And then just like with flash and freeze, you can then freeze your cells at whatever time point you want, 5 milliseconds, a second, 10 seconds, whatever you like. And so yeah, we call this zap and freeze. And really all this requires is just a few extra add-ons to your high pressure freezer. You just need this light stimulation system that shines the LED in like with flash and freeze. And then you just need a charging system that charges these little middle plates that you get that work much like any other kind of setup that you would have for high pressure freezing. And this enables you to electrically control your electrically excitable cells. And just a quick note here, all these things are very fancy and do seem, this is a real technical advance. But on the side of the experimentalist, it really doesn't add any extra difficulty to a high pressure freezing experiment. Really everything on my end is very much the same. All you have to do is just set everything up, you close the cover on the machine, and then it goes in. So there really is no added special technique on the part of the experimentalist to doing a zap and freeze experiment. In fact, I find this to be one of the easiest configurations to work with, despite the marvelous results that you'll see that we get out of it. So when we do zap and freeze, much as with flash and freeze, we can capture synaptic vesicle exocytosis and see vesicles that have fused. We can even do cool stuff like, for example, do serial sectioning, which Shigeki mentioned earlier, to get a 3D view of the entire synapse, and we can capture things like one synapse that has lots of different fusions all throughout them. So we can really capture these events very precisely. So now we come back to our question that we wanted to answer, which is what's going on with the dot vesicles? How does that relate to the synaptic output? And so now with this really precise temporal resolution, what we can do is we want to sort of walk forward and see what's going on millisecond by millisecond as this paired pulse plasticity sets in. Are the, as vesicles we know are depleted at first, are they actually coming back, or is it really just the remaining vesicles that are doing the work? So the way we do this is we walk forward every 3 milliseconds for our freezing time point. So our earliest time point that we freeze at, which I showed you there, where we capture exocytosis readily, is 5 milliseconds. There are just some physical limitations. Your temporal resolution is down to a millisecond, but essentially it takes a certain amount of time just for liquid nitrogen to actually freeze a cell. So for pure physical limits, 5 milliseconds is the earliest you can go. And then walk out from there as this plasticity sets in, and then also freeze after it at, say, 100 milliseconds or a second or 10 seconds after we know that synapses have gone back down to baseline. And so when we do that, here's what we get when quantifying docked vesicles. So as before, you can see that our vesicles are quite profoundly depleted by only a single action potential. And that's more or less maintained as we go forward until we get to 14 milliseconds. And it always is 14 milliseconds, where as if by magic, your vesicles rebound completely back to baseline. So the synapses, within this very brief time period, completely recovered all the dot vesicles. It's completely indistinguishable from our no-stim control. So this was really wild. This was not really what we were expecting to see, because this happened 1,000-fold faster than what Shigeki had previously seen with flash and freeze. And so the first thought was like, yeah, things are crazy different. What's going on? Well, it could be that this temporal resolution, again, we just missed it before, and everything else is the same. And indeed, when you freeze at further out time points, we get the same kind of results you got with flash and freeze. So if you freeze at 100 milliseconds, vesicles are actually back down again. And then as before, it takes about 10 seconds for the vesicles to again restore to baseline. So what we discovered with this technical advance with zap and freeze is that dot vesicle recovery at synapses actually occurs in two phases. The first of which is extraordinarily fast on the millisecond timescale, which we term transient docking, because those vesicles dock in, but then they pop off and undock, and then it takes a while for vesicles to again then be restored. And so this has become a really sort of hot topic in the field. There's a lot of things you can imagine about how these very fast vesicle dynamics can affect synaptic outputs, and this has all been really enabled by this system. So that's the end of the data. What I will say is that especially when we're talking about plasticity, many of the neuroscientists here might be saying, well, everything was just done with single action potentials. And that's true. We always tend to start with that simplest case of just stimulating once and then seeing what happens. But really, this system is amenable to any kind of electrophysiology experiment you could possibly design, any kind of train stimulation, LTP, LCD, any kind of weird protocol of stimulation you could possibly imagine with whatever amount of action potentials, with whatever time point. You can all do that with a high pressure freezer, and I have done things like that. I've done lots of complex kind of stimuli. All you have to do is essentially just program that into the freezer. You just use the touchscreen to tap it in, and then you can really look at whatever kind of process you can possibly imagine neurobiologically. And so, yeah, that's really what this time-resolved electron microscopy has allowed us to do. With this technical advance, we've discovered these new processes that had not been seen before, and we really hadn't been expecting, because nobody could look at these kind of things before, like ultra-fast endocytosis, transient docking, being able to directly visualize synaptic vesicle exocytosis and endocytosis. And so with this system, you can really do the same. You can say, I want to look at this mutant, I want to look at this simulation protocol, this plasticity paradigm, and then you just do your experiment. And the wonderful thing is you never know what you'll see under an electron microscope. I haven't talked at all about the postsynapse. There's lots of things you can capture there. You really never know what you're going to see, and you really will wait to see what you can get at this beautiful amount of resolution with this temporal component added. And so with that, I want to thank you all for being here and listening, and I want to thank everybody that made this work possible. Not least, particularly essential to my work were Sumana and Kie in the lab. And I also want to shout out Tyler, who's my good friend, and also the one other guy obsessed with docking in the lab. And also, of course, the wonderful engineers at Leica, as well as Wayne Davis, who all built these wonderful toys that I get to play with, and everybody and Laborers for organizing this. And with that, we'll take any questions.
Thank you, Shigeki and Grant, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, let's get started. Our first question asks, are there any artifacts introduced by high pressure freezing?
Okay, I can take this question. So this is actually quite a difficult question to answer, but I can tell you that there's some artifacts that you may actually get based on ice crystal damage, ice crystal formations. So when the ice crystal forms, then you can get membrane breaks, as I presented, and then you do actually see some evidence of that when you actually look through the images, and so you do get some sort of artifact. And so, you try best to avoid the ice crystal formations, but that could happen. But besides that, I don't think high pressure itself is actually doing anything to the cells while they're actually being frozen. We have some evidence of that, but nothing that we can share at the moment.
Great, thank you. Our next question looks like this one is for Grant. It asks, is it possible to test that the simulation is working with flash and freeze and zap and freeze?
Yeah, so for flash, this is relatively trivial in the sense is it's the same kind of controls you do for any other channel rhodopsin experiment. You know, you just have whatever your configuration is, whatever your preparation is, expressing channel rhodopsin. You know, do the same stimulation you do for flash and freeze, but do your EFIS recordings, the same kind of thing you'd normally do for to measure action potentials for channel rhodopsin. For zap and freeze, it's a little trickier because unlike flash and freeze, where you're just doing channel rhodopsin simulation, the field simulation is a specific configuration. So the zap itself, as it works in the instrument, only happens in this closed sandwich, in this closed thing that's not accessible to EFIS, obviously, because it's a closed system. You can't put any electrodes in there, and not really accessible optically to things like directly measuring action potentials. That being said, there are two ways around that we've gotten around that. So you can do some other methods. So we've stimulated cells, for example, directly in the with the system, and then done some less precise experiments just to check that you get activity. So, for example, you can do FM dye. So we did that. We stimulated them in the machine. Then instead of freezing them, we do FM dye. We fix them and then do FM dye imaging after that. I think other people have done stuff like calcium imaging to check that it's working. But if you want to really directly measure your action potentials and say, you know, I'm getting the exact pattern of action potentials that I want to, you can just use a field simulation configuration of the same type, you know, with the same field strength, that kind that you'd use for live imaging or EFIS recordings like that as a point of comparison.
Great, thank you, Grant. We have another question here. Looks like this one is directed towards you as well. This one asks, when do you use the flash and freeze method versus the zap and freeze?
Yeah, so I focused on, in my case, where zap and freeze was enabling as a technology over flash and freeze because of the added temporal precision. So obviously that's not going to apply to all experiments. You know, much of the time you won't be freezing at these kind of very, so if you want to look at transient docking, you got to do zap. That's as far as we know what we know right now. But for other things, especially if you're freezing at longer time points, everything looks little. For example, we've investigated ultrafast endocytosis with zap, and it looks just like it does with flash. If you're using the same system I was showing, cultured hippocampal neurons, zap just seems like the easier thing. You don't have to worry about channel rhodopsin. You don't have to do any of that. It's just a simpler system. Where flash can be really important is obviously it gives you all the kind of advantages that you get with channel rhodopsin in neuroscience generally. That is, if you're working in an intact organism or you're working in slices and you want to stimulate specific pathways and know what you're, stimulate specific neurons, then you're obviously going to use flash. So it's going to be good for a lot of systems like that. Because apart from the sort of things I discussed, the differences are mostly subtle. It's just going to be, yeah, what's the best for your system in terms of what you're trying to investigate.
Great, thank you. Looks like we have time for one more question here, and this is a two-part question. So this asks, can this high-pressure method be used for every type of cell, and can it get these beautiful images from every type of cell, or is there cell favorite types?
Okay, so yeah, and technically speaking, you can use this technique to look at any types of cells. The only issue is that there's actually a limit to the thickness of the tissue that you can freeze. And so typically this is supposed to be about 200 micrometer, and then that basically minimizes the ice crystal formation, and then you get pretty decent images, decent preservation of tissues out of those samples. You can use any types of cells, and then can you get beautiful images from every type of cell? So this is actually an interesting question because it really depends on the users, and so you have to really practice on loading the samples, handling the samples in order to get beautiful images, and so it takes quite a bit of practice. But once you get used to doing the freezing, I think it's gonna be, this technique is gonna work, and you can always get good images.
Great. Well, thank you again, Shigeki and Grant, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.